Good afternoon, everyone, and thank you for taking the time to join us today for another of our Saints Talks, short presentations and lectures by leading members of the academic community on subjects we hope are of general interest. My name is David Williams and I look after the alumni and supporter relationship with St Andrews, and I'm delighted to welcome you this evening and to introduce you to Dr Catherine Adamson. Dr Adamson gained her PhD from the University of Manchester and she's worked in the US for the National Institution of Health. Apart from the coronavirus, her other area of expertise includes HIV and RSV, which is one of the viruses responsible for the common cold. Dr Adamson's research was one of the casualties of the biomedical science building fire in 2019, which destroyed her laboratory and meant she had to start her research programme entirely from scratch. She is now the virology lead on several interdisciplinary projects tackling the coronavirus that causes COVID-19. Dr Adamson will talk for about 20 or 25 minutes and then she will take questions afterwards. If you want to ask a question, you can just do so by typing in the Teams taskbar. Try not to put your full name, as for data protection reasons, we try not to publish them. We will aim to finish about 10 to 6 Scotland time. And now over to Dr Adamson. Thank you.
Okay, thank you very much, David, and thank you for the opportunity to be able to talk to you today about our research that's tackling the SARS-CoV-2 virus, which is the coronavirus which causes COVID-19. Now, although I'm giving the talk, our approach has been very much multidisciplinary and collaborative, and there's many talented scientists working together on these projects. So the two projects that I'm primarily going to talk to you about today are with collaborations with scientists in the School of Physics at St Andrews. And I have an acknowledgement slide at the end, but I will try to point out key people as I go along. So the work I'm going to talk to you today is primarily focusing on what we're doing in St Andrews, but as all science, it's building on the shoulders of others' work, and there are many other scientists around the world that are taking related approaches, all with the common aim of combating this virus. So we come to SARS-CoV-2, the virus, the beast that's caused all of this disruption and loss in recent months. So I've chosen here to show you an artist representation of the SARS-CoV-2 virus. It's pretty much my only chance to get any kind of art or culture into a science talk. And this artist, I've got his webpage at the bottom here, creates these giant glass sculptures of viruses. And he has a whole microbiology theme, and if you go to his website or go to any of his exhibitions, you can view the likes of HIV, Ebola, smallpox, the bubonic plague bacterium that causes Black Death, and of course the coronavirus. So although it's an artist impression that we've got here, you can see the key components of the virus that we need to be able to understand and think about during the talk today. So inside the virus here, you can see the single-stranded RNA genome of the virus, and that is encapsulated by the viral envelope, which is studied by these protein trimers, which is the spike protein of the virus, which is the protein that interacts with a specific receptor on the human host cells, and it's that interaction that's essential for infection to start a new round of virus infection. So although this is a giant sculpture, glass sculpture of the virus, it's important to remember that viruses are absolutely tiny. So the SARS-CoV-2 virus is about 100 nanometers across. So 1 nanometer is a billionth of a meter, or to put it in context another way, the average cell, such as a red blood cell, is about 10,000 nanometers across. And as a virologist, I've always found it fascinating how such tiny, simple viruses can cause such impact in terms of disease, and the complexity required on our part to be able to combat these viral diseases. So in my lifetime, there's been 2 novel viral pathogens that have emerged and caused pandemics and caused millions of deaths. And it's quite sobering. When I first wrote this slide, I have to update these figures for the COVID-19 figures every day, and I updated these just yesterday. But the first pandemic in my lifetime was the HIV/AIDS pandemic, and really I spent much of my scientific career working on this. So I primarily worked on a novel class of HIV inhibitor called maturation inhibitors. And you can't just start working with HIV, you have to work with it within a biological safety containment facility, BSL-3 or category 3, which is a specialized facility that enables us to contain the virus for safety purposes. And I have over 15 years experience of working with viruses in category 3, primarily with HIV. And I used this experience. I felt a really strong sense of duty as a virologist, aside from the fact that my entire lab burnt down, but I felt a strong sense of duty if I could do something to help towards the COVID-19 pandemic way back in March when it all started, that I should. And the one skill that I had that was unique in St Andrews is this experience of working with highly pathogenic viruses at category 3. And so I could bring the ability to work directly with COVID-19 to St Andrews to work with the virus, and therefore allow research on this virus. So like HIV, you can't just start working with SARS-CoV-2. You have to gain permission from the local university, but more importantly from the National Health and Safety Executive. So I had a conversation with the director of the Cat3 lab at St Andrews, Professor Terry Smith, and we started to put together an application to the Health and Safety Executive, which was approved in April of last year. So now I've got approval to work with the virus. The next thing I needed to do was actually obtain a purified isolate of the virus to actually work with. And you're in the middle of a pandemic and you're surrounded by the virus and everyone's in lockdown. But actually this was much more difficult to do than you would actually think. Not because there wasn't a scientific will to share the virus, but mostly because you had to find someone in all the crazy, all the virologists were super busy. So you had to find somebody who had time to read your email request, and more importantly, had the time to actually package up the virus and ship it to you. And I got lucky after sending several emails to Public Health England. There's a postdoc, Dr. Marion Killip, and she used to work as a postdoc in Professor Rick Randall's lab at St Andrews, and she'd been working in Public Health England for several years. And she responded to my email request and kindly shipped us the virus. So that got us started, and the virus arrived in St Andrews in May. So then my next task is to actually start working with the virus. So I have to go into the Cat3 lab, and I'm used to working with HIV. So that's a bloodborne pathogen. It's relatively very challenging, I would say, to actually infect yourself with HIV when you're working in the Cat3 lab if you remove all sharps. Working with a respiratory pathogen like SARS-CoV-2 is a step up. It's a step up for me, where even just one drop in the wrong place, you could cause an infection. Also, my first job was to go into the Cat3 lab and basically try to make as much virus as possible and as concentrated as possible, which I can tell you takes some considerable reverse psychology when I, like the rest of the population, was in lockdown for about 8 weeks, doing everything I possibly can to avoid the virus. And now there I am trying to make as much and as concentrated as possible. Well, I got beginner's luck and I made a decent high titer stock. And so now I have a stock of virus to work with. So the next thing we needed some money to actually perform any research. So we started writing grant proposals, which is the way it works in science. And the first grant proposal that I got funded was from Medical Research Scotland, which is actually a project to screen for novel compounds that have the potential to be antiviral drugs in the future. And I'm not going to talk about that project today, but several other, we continued on the grant writing, and I have to say it was a bit different to the way I normally write grants. So all of the grants were written collaboratively, mostly with people I've never met before. I had never worked with them before. And in the case of Peter Wall, I'd never even spoken to him. We simply fired emails back and forth in a frenzy of grant writing, and basically got the grants written and submitted. And this approach seemed to have been quite successful, and maybe I should adopt it as I go forward rather than my usual procrastination and talking myself out of it. But I was involved in writing 9 grants, 7 of which were funded, and we raised a total of about £600,000. So we could start our research. And the 2 projects I'm going to talk to you today, the first one was funded by the UKRI COVID Rapid Response Initiative, and we started work on that project in September. And then the second project was first funded by Scottish Funding Council SAF money which was distributed via the University of St Andrews. And then very recently we got top-up money for that project from the Department of Health and Social Care Innovations and Partnerships Fund. So I've got money now, so I can start the projects. And the 2 projects I'm going to talk to you about today really are about preventing transmission of the SARS-CoV-2 virus. So this virus can transmit in 2 main routes. And all the things that we've been doing during lockdown, following restriction measures, are all public health measures to prevent transmission of the virus. So wearing masks, washing your hands, social distancing, it's all preventing transmission of the virus. And this virus can transmit in these 2 main routes. So you can have, here's a person coughing and sneezing, and when that happens, you can have the virus in tiny droplets, which remain aerosolized, so they're in the air, and therefore you can breathe them in and get transmission in that way, particularly if you're in indoor, not very well ventilated environments. And then the second route of transmission is slightly larger droplets that are emitted from the coughing or the sneezing person. And those droplets containing the virus can fall onto surfaces. So here's a desk, and then you can touch those surfaces. So you pick up the telephone, and then you can transfer those viruses to your mouth or your nose and get transmission that way. So the virus has to get access to the live cells inside your mouth and nose to have transmission. So our aim of these projects is, can we develop new ways to stop this transmission that are physical methods, that are behaviour independent interventions? In other words, they're sort of there in the background and maybe you don't notice them, and you're not relying on the person to choose to take part in that intervention. So do they choose to wear a face mask? And you're not relying on the person to do it correctly. So even if they've chosen to wear a face mask, are they wearing the face mask correctly? So it's try to get these behaviour independent mechanisms. So here are the 2 projects. So the one at the top, the first project here, is primarily focused on these aerosolized viruses in the air, but actually this technology could be used to actually inactivate viruses on surfaces, but we're primarily focusing on the viruses in the air. And then the second project is working on these viruses on surfaces, so suppressing SARS-CoV-2 transmission through surface engineering. And I'm going to talk about this project first. So the project's a collaboration with Professors Peter Wall, Philip King, Andrea De Falco and Terry Smith. And what we're trying to tackle is this survival of the SARS-CoV-2 virus for multiple days on commonly employed surfaces, so on stainless steel, on plastic. So think about your door handle in an office or the plastic of a telephone, etc. And we want to break this viral touch surface transmission pathway through the identification and development of surface materials or coatings so that we can inactivate SARS-CoV-2. So if it inactivates quickly on the surface, we've got less chance of touching it and passing on the virus to ourselves. So our overall goal is to identify antiviral surface coatings that can be easily, rapidly and cheaply deployed in public places or on personal items. So I've given examples of door handles or elevator buttons or telephones, but also on personal items such as mobile phones, keyboards, tablets, etc. And we'll do this by building on existing knowledge about antiviral properties of copper. And like I say, all science is generally built on the shoulders of others' work, and we're building it on the antiviral properties of copper. So why copper? Well, if you work in the lab and you culture cells, which we need to do for virology, you need a tissue culture incubator, and you can buy those incubators with a stainless steel interior, or you can buy them with a copper-lined interior that's shown here. Now, most people have the stainless steel version because it's cheaper, but if you invest your extra money into your copper-lined incubator, I have a copper-lined incubator in the Willie Russell labs. I think I'm the only one who bought a copper-lined incubator. I got a fairly good deal, so I didn't really spend any much extra money. But the real advantage is the antifungal, antibacterial and antiviral properties of copper. So the last thing you want is bacterial contamination in your cell cultures. So it's there to prevent contamination, and it's also supposed to reduce the burden of continually cleaning your incubator because of the antiviral properties of copper. So that's an example of the use of copper for infection control in the lab. How about in other settings where infection control is even more important, such as hospitals? So in areas in hospitals where infection control is really important, copper is already used. So here you see in a hospital a copper handrail, for example. But this is relatively rare and in specialised environments. And if you think about it, actually stainless steel is everywhere. So, you know, your door handles, rails in public places, in railways, stations, on your elevator buttons, etc., etc. So can we develop a technology that we could just coat these stainless steel surfaces easily and cheaply with a surface that has antiviral properties? And can we even go beyond copper and try to develop novel surfaces that instead of copper are clear, so that we can use on mobile phones or keyboards? I mean, you can't spray a layer of copper over your keyboard, you wouldn't be able to use it, but maybe we can develop novel surfaces that have clear properties or properties that are suitable for different applications. So the antiviral activities of copper have been shown by numerous groups with different viruses. And here I'm just showing our data that agrees with others that, in fact, if you use copper surfaces, you can inactivate SARS-CoV-2. So what we have here is we've got 4 test surfaces, we call them coupons. And we've got our 2 controls, so no coupon at all, so it's just virus as it normally would be. And over time, with the no coupon control, you don't expect to see significant inactivation of the virus. So you've got 100% survival across this time range. Then we've got our stainless steel control. I've talked about stainless steel, and you can see we do get a bit of a drop in virus survival over time, but it's nothing like the 2 copper samples. So here we've got 2 different types of copper. We've got copper foil, which is a solid piece of copper, and then we've got evaporated copper, which is using the technology that the physics that we're collaborating with use. So you take a coupon, which is made of a inert substance, so a substance where you don't get any inactivation of the virus, such as stainless steel, and then you put a thin layer of the copper over the top of it. So that's the evaporated copper. And you can see both the copper samples work in just about the same way. So at 30 minutes, you'll see about 50% inactivation of the virus. At an hour or 60 minutes, you're going down to about less than 20% of the viruses survive. And then at 120 minutes, we see no survival of the virus. And this little dotted red line here just shows the limit of detection of the assay that we're using. So that's good evidence that copper, fantastic, inactivates virus. Maybe need to leave it for about an hour. So obviously we can improve on that, hopefully. So you might be interested in how we actually get that data. So remember, everything has to be done in the Cat3 lab. And I have a very talented postdoc, Dr. Jane Hilton, working on this project. And here Jane is working in the Cat3 lab, performing these experiments. So as I say, we use these test surfaces that we called coupons. They're about 4 by 4 millimeters. So they're very small. This is an example of a copper one. We need to keep everything sterile because we need to use cell culture. So the coupon comes sterile. We use tweezers to transfer the coupon into the well of a 96 well plate tissue culture dish, which is standard we use in the labs all the time. We have a stock of the SARS-CoV-2 virus, which I made in the first instance, and Jane's made new stocks. And we know how many infectious particles per milliliter are in this virus stock. So if we add 10 microliters of the virus stock onto the coupon, so here we have it here. So here's the copper coupon sitting inside the well of the 96 well plate tissue culture dish. And here's our 10 microliters of copper of virus. And because we know how many infectious particles are in our stock of the virus, we know exactly how many virus particles we're using to challenge the copper with. So we leave the virus on for the various time points. So in the data I showed you, it was 30, 60 and 120 minutes. We then recover that virus by pipetting up and down into a tissue culture medium. We then take that and we put that virus onto mammalian cells that can support the SARS-CoV-2 virus replication. And we're using cells called VeroE6 cells. And here's an image of VeroE6 cells. And what we have is what we call a confluent layer of these cells. So it's a complete blanket, if you like, across the surface of the tissue culture well. So there's no gaps in the monolayer. The cells are all tightly packed in, no holes in that monolayer. And you can imagine that a virus comes in and infects one of those cells, okay? And then what we do after we've done the infection step, we put like a semi-solid support on top of the cells. So the original cell that's infected with the virus will produce new viruses because we incubate it for 3 days. But the new viruses can't move far away from the original cell that's infected. So if you like, it's a local area of infection. And when the virus infects these VeroE6 cells, it completely kills the cells, it wipes them out. And so you can imagine what you get is for each spot where an individual virus infected, your cells are all killed. So what you get is a hole in that monolayer. And we can see that by staining up the cells. So living cells, we use a stain called crystal violet, and those living cells will all stain up blue. So here at the top, here's a well. All it's got in it is VeroE6 cells. There's no virus, and you just see this blue monolayer all the way across. And then the 3 below show increasing numbers of virus particles we used. So you can see these white dots are the holes in the monolayer that we call plaques, where the virus has killed the cells. And we can count these plaques, and therefore we can know how many infectious particles were there before we did the exposure to copper and after the exposure to copper or different surface. And that way we can get that data that shows how efficient the different surfaces are at inactivating the virus. And you can see there's about 10 plaques on this one, about 50 here, and here there are so many virus infections that all the holes have merged together, so we can't count the plaques. So that's how we get the data. So what about the novel surfaces that we're going to test? Well, to start off with, we've divided them into 3 main groups. So we've got elemental metals, metal oxides, and nanostructures. And we're using this technology in physics, the ability to really precisely cover surfaces with layer upon layer of the different test surfaces, and also the ability of the nanophotonics facility to incorporate textures or different structures into these surfaces to see if that can inactivate the virus. And all of these test surfaces have been generated by Makio, Yoshiko and Mai-san, which are postdocs in the physics groups. And let's go back to the data that I showed you originally. So how are we going to be able to test these test surfaces to see if they are better than copper? Well, what we really want is this time point here at 30 minutes, because at 30 minutes with the copper, we've got about 50% inactivation. So that gives us a window to be able to see a test surface that inactivates quicker. If we try to do it at 60 minutes, we wouldn't really be able to see anything that activates quicker, and we won't see anything at all at 120 minutes. So it's this 30-minute time point that we're going to use for screening different test surfaces. And here's a hypothetical scenario, okay? So you've got your evaporated copper, does as we expect. At time 0, you've got 100% survival. At 30 minutes, you've got about 50% inactivations. This isn't real data, we're just making it up to show you the scenario. And then we've got our two controls, stainless steel and the no coupon, which we wouldn't expect to see any inactivation. And then we've got three hypothetical test surfaces. And what we really want to see is what we see with test surface 1. So we get significantly better inactivation than copper. So this would be the dream. Test surface 2, this is not what we're looking for. It's no better than copper, don't get very much inactivation. Test surface 3 might be more realistic what we might see, but actually if we see something like this may well be something that's worth pursuing in the future for further optimization and tests. So even if it's equivalent to copper, that test surface may have properties that are desirable, might be cheaper, might be more durable, might be clear so that we can use it on mobile phones, keyboards, etc. So even if it's equivalent to copper, we're still interested in it. So we started testing elemental metal surfaces. We tested 4 so far. So we tested silver, bismuth, nickel and palladium. And you can see following that hypothetical scenario, we have got a little bit of inactivation. It's definitely not better than copper and it's not much better than the control. So we're not going to follow these 4. That's what we did first. And then we looked at metal oxides, and we have various different metal oxides that are listed here, and you can easily see that these two, copper oxide and cuprous oxide, have got activity that's as good as, even if a little bit better than, just the straight copper. And this is good. It tells us that our assay is working. Other people have already published that these two oxides also have activity that's equivalent to copper. So it's reassuring that our assay is working as we expect, and we're also building on and confirming results of others. So that was good news. And then we get to the nanostructures, and we've tried 3 structures so far, and they're labeled texture 1, texture 2, and texture 3. And hopefully you can see straight away that texture 1 seems to be giving us some promising results. Okay, so we were very excited about this. We've repeated this experiment 3 times and we get the same results, so that's good, in 3 independent experiments. And it's not as good as copper, but it certainly looks very promising. And given these are the first 3 textures that we've looked at, we can build on understanding the way that the virus is being inactivated, which we don't know at this current time. It might be trapping the virus in the structure. The structure has sharp textures, so it could be bursting the virus or damaging those vital spike proteins on the outside. But this is certainly something that we're following. We could combine this texture. So the texture is made simply from silicone, and you can see silicone, which is just a flat surface of silicone, doesn't have any activity. So it's something to do with this texture that we've incorporated, this nanostructure that's having this inactivation activity. So we could combine this silicone texture with a layer of copper, which is what we're trying to do now, to see if we can get that activity down to the same as copper, or hopefully even better than copper. Now, I can't tell you what these textures are, because obviously this is a promising result and this is unpublished data, so please don't ask me that at the end. We simply can't disclose it as unpublished promising data at the current time, but we're really excited about it and we're moving forward with this. So that's where we've got to with that project. I'm going to move on to the second project, which is St Andrew's Light technology, which we cunningly called SALT. I think that was Kenny's idea. I can't claim credit for it. And as I say, we're primarily looking at the activity of this technology inactivating aerosolized virus, but it does have the capacity to also inactivate surface virus particles. But we're going to focus on the aerosolized portion. So this is a project, there's now a huge group of collaborators on this project, which has been spearheaded and started by Kenny in the School of Physics and Ewan, who works at Ninewells Hospital. And I'm the virologist on the project, but they really have spearheaded and brought together a really exciting group of many people, which I will hopefully list all of them in the acknowledgement slide at the end. Our primary aim here is to use a multidisciplinary approach, hence the need for so many collaborators, to investigate inactivation of aerosolized viruses using filtered Far-UVC light. I'll explain what that is. And the safety of such technology in occupied places. In other words, can we use that technology in a lecture theatre that's full of students? Can you use it in crowded places where chances of transmission are high? Okay. And our overall goal is implementation of this technology in public spaces. That's what we would dream to do. And we will do this by building on existing knowledge about the antiviral properties of UV light. So again, we're building on scientific knowledge that stretches right back to the 1800s, actually, for UV light. So the mechanism by which UV light inactivates viruses is much more clearly understood, at least for nucleic acids, than the copper or the nanostructures that I talked about before. We really, we're hoping to get more money to investigate the mechanisms there so we have better understanding. But how UVC light inactivates viruses is much more clearly understood. So UV light strongly absorbs nucleic acids. So when I say nucleic acids, I'm talking about DNA or RNA. So we have a genome that's made up of DNA. Viruses can have a genome that's either DNA or RNA. The SARS-CoV-2 virus has an RNA genome. And when the radiation from the UV light is absorbed by nucleic acid, it causes damage. So it can cause breakage in the bonds of the basis of the DNA, which is illustrated here. And you can get, for example, a common breakage is and like formation then of these pyrimidine dimers. So you're causing damage to that nucleic acid. And if you can imagine a whole genome of a virus, it doesn't happen in one place. It happens in multiple places across the genome. So you've got significant damage to the genome, and the genome is needed once the virus infects the cells. It's the instructions for how the virus replicates itself. So if you've damaged the genome, you can't make new viral proteins, new copies of the viral genome, and assemble particles. So it completely inactivates the virus. This UV light is also absorbed by proteins, particularly at lower wavelengths, and so you can damage the proteins of the virus as well. So I told you about these spike proteins that are essential for virus inactivation. If you damage those proteins, they're really conformational dependent. So if you damage them, you're not going to be able to have a virus that can infect cells, and therefore you've inactivated the virus. So when we're talking about UV light, what we're talking about, so this is the electromagnetic spectrum, and we're interested in UVC light, which is between 200 and 280 nanometers, okay? So we're talking about UVC light, and the most common source of artificial UV light, so how we can make it, are through these low pressure mercury lamps that emit at the wavelength of 254 nanometers. There are also some LED lights coming on stream now, but it's really these UV lights that are being used commonly for inactivation of viruses, bacteria, etc. So one application, again, if you were working in the hood, so this is the type of microbiology safety cabinet that you saw Jane sitting at earlier that we use to do all our virus and cell culture work. It creates a sterile environment, and it also protects the worker from exposure to the virus. So that's why we use these hoods. And after we've finished working, we inactivate. So we've cleaned everything up, we've tidied everything up, we close the hood, and then we switch on the UV light, and that runs for about 20 minutes, and that's a deactivation step. So anything that might have been in the hood, any spilled virus, which shouldn't be there, but everything should be inactivated. So that's one application that is commonly used for UV light. We also use UV light, and you may have seen this on the news, to inactivate spaces, industrial spaces. So there's UV light emitting robots that go around airports when people aren't there. You use these UV lights as shown in this picture here to sterilize a hospital room with no patient in it. And generally these UV lights are used in areas that are not occupied by people. So it's quite well known that you can get erythema, which is basically sunburn, from UV light, so it damages the skin and damages the eyes. If you get the damage to the eye, it's kind of equivalent to snow blindness. And also, I've just explained to you how you can damage the genome, and you can damage the genome of cells, so there's a risk of skin cancer, etc. So these low pressure mercury lights are typically used in unoccupied places, although there has been experiments that have been done since the 1930s or even earlier, where they're using these UV lights at 254 nanometers in what's called upper room germicidal UV inactivation. So what this is they take lights and they fix them to walls or ceilings. You can see them here circled in this picture from the 1930s, and they emit the UV light, but in an area above, so it's like this disinfectant zone above the people's heads. You have to have quite a tall room to be able to do this. And you've got circulation of the air. So the prediction is the viruses will move into this disinfectant zone above people's head and get inactivated. You would have to be really careful. You couldn't have people being able to reach up into this zone, etc. But it has been used. So in the 1930s, Wells used it to reduce transmission of measles in Philadelphia day schools. And measles is one of the most infectious virus known. It's been used to reduce spread of influenza in the 1957 pandemic, and it's been used from the 1950s even to the present day for preventing TB transmission. But obviously this is not ideal, and really there's a resurgence now in the interest in UV light, and this is what we're interested in our project, is what we called Far-UVC, which is at 220 nanometers. So at a lower wavelength than the lamps I've already talked about, okay? And at this wavelength, it's predicted, and there's quite a lot of data now, and I'll talk about it in a minute, that actually this type of UV light, as long as it's filtered, so they come with an optical filter, so it's only emitting wavelength at 222 nanometers, that this light is safe to use in human occupied spaces. So this is where the interest is coming in. And it's called Far-UVC light because it's a lower wavelength away from these more conventional, already in use UV devices. And so I've said that it's proposed to be safe to be used in humans. And the primary reason for that is it's actually got, this Far-UVC light has a very low penetration. It only penetrates about a few millimeters into the skin, or if you're thinking about eyes, into the eye. So if you think about skin, you'll know that the top surface of your skin is actually dead skin that sloughs off all the time. That's primarily what dust is made of, etc. And the UV light only penetrates into this layer and it can't get any further, whereas the conventional 254 light can get down into the epidermis layer where the living cells are. So if the light, the Far-UVC light, can only get into this dead layer, it can't damage living cells, it can't be a cancer risk because it's only getting into the dead layer of cells that's going to be sloughed off. And there's quite a lot of experimental evidence in lab now and in animal models that support this hypothesis. There's more for skin than there is for eye. So for the eye, it's the tear layer that protects your eye from any deeper penetration into the important cells in the eye. And I've just shown this data from a paper in 2018. This is not our work, but you can see with the 254 nanometer UVC, which is shown along the top here, these hairless mice have got like red patches of like sunburn, which is one of the symptoms of being exposed to UV light. But if you look at the Far-UVC 22 nanometer, there's no evidence of that going across. So I went to a webinar last week, which was a panel of experts on Far-UVC set up in the United States. And the consensus from that meeting was they're quite happy with convincing evidence from the lab and animal models for skin. A bit more work needs to be done on the eye, but all the data on the eye seems to show the same thing. But we really do need clinical studies. And our collaborator at 9 Wales Hospital, Ewan, got parallel funding to our Department of Health and Social Care Innovation Fund funding to conduct skin safety clinical trials with volunteers at a 9 Wales Hospital. So that's underway. So that's exciting. So I just wanted to show you some data. This data was generated by Wenja, postdoc in the lab who's working on this project. And she's starting working with influenza virus that we can work at category 2. And the reasons for that will come clear as I show you the next couple of slides. But you can see here, these are exposures of a few seconds with the UV light. And you can see even with relatively low doses, you can get 99 or 100% inactivation of the influenza virus. We're using the same plaque method, but with influenza virus to quantitate the viruses after they've been exposed to UV. And this is some pictures that she took in the lab of the lamp that we're using. So you can see here that what she has is the influenza virus in drops in a tissue culture and she's exposing it to the light, calculated how long and how far to get the dose. And then we're getting the data to show that you get inactivation of the virus. And this has been shown for many different viruses. This is just our data. But I started at the beginning saying we want to use this technology for looking at aerosolized virus. And this is not aerosolized virus. But how do we do that in the lab? If you mention to a virologist, I want to work on aerosolized virus, they would be like, no, 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 no. When we're trained to work with viruses, particularly in the Cat3 lab, everything that we do is to try to prevent creation of aerosols. You don't want to be spraying pathogenic viruses around. So how do we go about looking at the effect of UV light on aerosolized viruses in the lab? Well, this is a contraption that we're having built. So it's an aerosolized virus chamber that ultimately will go inside the tissue culture hood. And we're collaborating with Professor David Brenner at Columbia University. They've built a similar aerosolized chamber that they've used in the States. They've used it for Far-UVC light, and they've shown this to look at the effect of UV light in aerosolized influenza and other coronaviruses, but they don't work in category 3. So our hope is we can eventually get this into category 3. But we'll be starting with influenza virus that can work at category 2 for safety reasons. So the guys in the physics workshop, Callum and Mark, have been building this for us. So basically what you have is you have a nebulizer here that creates the spray of virus. So it's a liquid culture of virus, and then we spray it and it goes into this sealed chamber, sealed being the important part here. So it's not spraying around the tissue culture hood or anywhere else. And it has a window to expose your UV light. So you would put your lamp up against that window and expose it for the amount of time you need. The virus end, there's an air pressure coming through here. And the virus that's left would come, and we have these gelatin filters that fit in this sample holder here. And the viruses will stick to the gelatin filter. We can then remove it, dissolve it in tissue culture media, and then go on and quantitate our viruses using the plaque assay. We're also hoping to get a biosampler that we can take a sample of the air that contains viruses. And then any virus that's not collected by the biosampler or the gelatin filter then comes out this way and then gets bubbled into liquid waste, which is disinfectant to inactivate the virus. And Callum has, worked very hard on this and built in an emergency overflow control system. So we have a safety system, so if the pressure builds up, it's not going to explode, but we are going to try it out with a category 2 virus in the first instance. We'll probably just try it out on liquid first. And this is almost there. The pieces of equipment that we need that are in the blue boxes are on order, but they haven't arrived yet. So we don't actually have this in our hands yet, but we're building it. And this will allow us to study aerosolized viruses in the lab. But that's still in the lab. That's not real, like, life scenario, what happens in a room. And so we're trying to get closer to real life scenarios, and you can do that by computational fluid dynamic simulations. Please don't ask me any questions on this. I really am not working on this. This is the area of expertise of Kenny in the University of in the Physics School of Physics, and he's actually an astrophysicist, so it's all complicated calculations that are way beyond me. And he's working in collaboration with a company, Fluid Gravity Engineering, which provides some of the software. And they're looking, so this is, they're taking a typical classroom. So this is room 203, a lecture theatre in physics, and they're doing simulations on ventilation of air, virus particles, UV doses, and trying to model what would happen in a room scenario. But that's not actually real life, what would happen in an actual room of people. And Kenny did ask us whether we could do this in the Cat3 lab in St Andrews. But obviously the idea of spraying SARS coronavirus around our category 3 lab was, like, to me, horrifying. And that was a no. And it's just like, well, I don't know how on earth we would do that. And then I got an email from Kenny. The title was In a Basement in Leeds, and which was rather ominous. But basically, so that there is a facility, I don't know, there may be more, in the UK that you can do this. And it's an aerobiology test room in the University of Leeds. It's run by Professor Kath Noakes, and we now have a collaboration with them. So the room is an average sized room that's similar to a hospital room, and there's a collision nebulizer that basically you can use to spray in your test microorganism. And we're going to go down and install our UV lights, and then you can imagine doing experiments without lights, with lights, different doses, how are the virus inactivated. And the way we would come into this is we want to eventually try it with our influenza virus, which is why Panja is working with the influenza virus. This is a category 2 facility. You can't work at category 3. We can't do this with the SARS coronavirus, but we can do it with the influenza virus. And then we would provide the virus. Wanja's made a stock of 120 mils that we can ship of the virus to ship down to Leeds. They then perform their experiment, collect the virus, ship it back to us on dry ice, and then we do the quantitation. So that's where we come into this. We haven't started this yet, but we do have the funding in place to conduct these experiments. So this is rather fantastical. And if you'd asked me a few months ago that I would even be talking about spraying pathogenic virus into whole rooms and looking at the effect of Far-UVC light, I would have told you were crazy. But here we are now. That's what we're doing. So this is just a quick summary. I've talked about tackling coronaviruses on surfaces in the air. Our approach is behaviour independent, and it's broadly acting. So all of our approaches would work on the different variants. It works against a broad range of viruses and bacteria, and our ongoing research is therefore important as it has potential to tackle really big issues in infection biology, such as multi-drug resistant bacteria and the next pandemic virus when that emerges. And this is just our acknowledgement slide, and I think I've gone a bit over time, a bit over enthusiastic, so I apologise for that.
Just to say thank you, Dr. Adamson. You know, we often use, when we're talking to university audiences, we also often use the phrase cutting edge research, but this is very much the first time I've seen anything that tells us about what's going on in a way that affects everybody in the world. So tremendous privilege to be able to listen to you. I'm going to extend the questions to around 6 o'clock, just to give people time, as I'm sure there will be a number. But just as a starter, why does copper work? How does it work?
So we don't have a clear answer to that. And we're trying to apply for funding now to be able to really answer that question, the mechanism in the way that copper works. It's something to do with the copper ions dissolve, if you like, out into the solution of the when you put the virus on top. And there's been numerous proposals. It damages the nucleic acid, it damages the proteins, it damages the lipid layer, it produces reactive oxygen species, but really the answer is not known, and much more research is needed to be able to really nail down what that mechanism is. And if we could do that and understand how copper works, then we would be able to have a more rational design of the surfaces that we're using. So it's still an open question.
It's an anonymous question on the nanostructure, the first of the three. Was it created in-house?
Yes, and that's the beauty of the team that we have. You know, if I didn't have the collaboration with physics, I could try to purchase, you know, copper foil surfaces, so like the solid copper, and numerous of them are available, but that's as far as I'd be able to get. But the unique skills of the physics team, they are able to create all of those novel structures in-house, and that's how the multidisciplinary approach works, and I think that's what really gives us an exciting edge with this project.
Comment from Tim, really interesting and well explained even to a complete non-physicist. We do have a physics question, and I know you suggest stay away from them, but just in case, how does the inverse square law for light intensity impact the practical efficiency of UVC sanitation in large spaces, or is that something you feel is slightly outside your area?
So it is slightly outside my area, but the only thing I can tell you is, that's why we need to do the experiments to test the different doses, to test the spread of the light. The only thing I can tell you is there is currently a legal limit for Far-UVC over a period of 8 hours, and that's 23 millijoules per centimetre squared. There are moves in the US and the UK regulatory bodies to start increasing that dose. So it's probably not answering your question, but we're experimenting with dose and impact and all of that.
And I suspect a related question, anonymous question. Do these nanostructures have colour, and are they opaque?
Well, it's silicon, so it's just whatever silicon looks like. They're kind of, when we image them on the SEM, they look grey. We're not deliberately incorporating colour into them. It's just the colour of the material that we're using.
How long do you anticipate the research on both these streams to continue?
Well, at the current time, unfortunately, we only have funding that's going to last us to the end of the year. So the funding for the copper project runs out at the end of August, and the funding for the Far-UVC we have funded now until end of November. Obviously, we want to continue, and the grant writing frenzy is starting again, and we hope that, with encouraging results, we can secure more funding. But at the current time, it's just until the end of this year, if not earlier.
And a prosaic, well, she describes it as a prosaic question from Hillary. Given the life of SARS-CoV-2 on various surfaces, should I still be sanitizing or quarantining my groceries in case someone before me has put their hands on them?
If you were being really strict, yes, I would advise it. I did that at the beginning of the pandemic, and then I must say I've become a little bit more relaxed. So the most important thing is not your groceries, it's your hands. So if you touch those groceries and they have the potential to have the virus on them, as long as you wash your hands, because it's your hands that are going to transfer the virus to the nose and the mouth, where the living cells are that the virus can infect. So I wouldn't say you need to quarantine your groceries, which I do admit and say at the beginning I did that also, but what the most important thing is washing your hands. Your hands are the vector for transmission.
Another anonymous question. Are you working, is there any difference between variants of the virus in terms of these two modes of transmission? And are you working on any of the new variants? Are you working with the original coronavirus?
So that's one of the real positives of these two technologies. We don't envision that a virus with different variants, different mutations, would be affected any differently from the original variant. So it has that advantage that if you get a few mutations that has a significant effect on transmission or resistance to the vaccine, these technologies will still work because it's kind of like a broad effect. If you think UV light, it's inactivating across the viral genome, inactivating the proteins. So that's an advantage, in our opinion, to these technologies. We're not, being subjected to problems with variants. We only have the original variant in St Andrews. We did discuss it, but because primarily what we're doing, having a different variant wouldn't make a difference. At the current time, we are not growing any of the different variants in St Andrews. We don't have an experimental advantage to do so. So why would we start working on a more transmissible virus and increase our risk when we're working in the Cat3 lab? So we can't justify it. If we have a project where we do need to work with those variants for a biological reason, then we could bring them in. But at the moment, we're not working with the variants.
Another anonymous question. Can you give us a sense of how many other research groups across the world are working on these two areas?
I don't know. So you know, people maybe are established in these fields a bit more than we are, may not know that we're working on it because we haven't published anything yet. So you can never say. But all science, as I said at the beginning, is built on the shoulders of others. There are definitely multiple other groups across the world that are working on similar approaches. And that's the way that science should be, you know, because everyone's going to make slightly different observations, do something novel, confirm each other's results. So there are multiple groups working on the same technology across the world, and that's the way it should be, I think.
Indeed, another question from I presume another researcher at Florida Tech named Andy Kay. Have you studied the structural integrity of viral particles after exposure to the copper surface?
We plan to do that. I haven't got any data to show you in this talk today.
And then a more consumer-orientated question: are face masks with copper strands in them worth having?
Yeah, so we're not working on that, but there is a whole field of research where they're putting in these antiviral surfaces and into protective equipment, etc., building in copper and things like that into something like a face mask. So that is being worked on. It's not something that we're looking at.
I'm just going to move to a final couple of questions. A clever question from Trevor. Brass door handles are used in domestic situations. Brass is an alloy containing copper. How effective are they in antiviral and antibacterial stuff?
Haven't tested it directly, but we would think that they would have. I think brass tends to be less common than stainless steel because it's not as cheap. So, you know, in our building, we don't have any brass door handles, but maybe in domestic homes, you're more likely to see brass. So I think it would have activity, but I don't think it's as common as stainless steel, just based on cost.
And a final question from Hugh Graham. We've looked at ultraviolet technology in line with air filtration for our museums, but decided not to use these products due to the lack of evidence that it disinfects the air to allow us to give guarantees to our visitors. How do we get to the guaranteed point, and will your work in conjunction with Leeds give people this?
It'll be a step towards it. I think this is I think there's a lot of potential around this technology, but we do need to, as a field in general, gain more real world data so that there is convincing evidence out there. So the work in the chamber at Leeds will be a step towards that. I think it's quite difficult to imagine a scenario, and this could be a limitation how we do clinical trials, because you can't really release virus, put people in a room and then release virus and turn on lights. But that's what maybe clinical trials will be approved under certain circumstances with certain biological organisms so that it can be tested. So some people are not working with pathogenic viruses. Some people are using phage, which are viruses of bacteria, as surrogates, because the technology works on any virus because the virus is so small, the UV penetrates. So it doesn't really, it's always best, in my opinion, to do the closest to the real world scenario, but you can use phage, and we're planning on using phage as a surrogate before we get to influenza. So maybe the surrogate work with phage might get us closer to the kind of data that you're looking at.
Thanks, Dr. Adamson, and we're going to, with your permission, of course, publish your email address in the chat as well. So we have a few more questions left over, but I'm going to move to a close now. I'm also going to say we're also going to put our survey in the chat bar. It only takes a couple of minutes to complete. So if you do get a chance to fill that out as well, that would be very helpful. In 2 weeks' time, we're going to be joined by Dr. Jeffrey Mura, who is a senior lecturer in collective violence and an expert on the formation of identity in the context of conflict. So if you enjoyed this Saints Talk, we have a Saints Talk every 2 weeks up until the early summer, and you can watch our previous Saints Talks on our YouTube channel on the Alumni website, or you can follow us for news of them on social media via Facebook, Twitter, LinkedIn and Instagram. And now just to return with my thanks to Dr. Adamson for her time and sharing her expertise with all of us. Thank you very much for joining us.
Yeah, thank you for listening, everybody. Thank you.